So hello everyone, my name is Michael Ray. I am science writer here at SENS Research Foundation, and I am joined this morning by Dr. Amutha Boominathan. She is the head of our MitoSENS lab working on the repair of age-related damage to our mitochondria, which are energy-producing organelles in our cells. Dr. Boominathan, good morning.
Thank you, Michael.
Hi. So what's the big problem you're trying to solve?
The work in our lab is centered around maintaining the fitness of mitochondria over age or time. As you know, mitochondria are these important organelles that convert the food that we consume into energy. They are also implicated in several important functions in the cell, including hormone and calcium balance, immunity, and in controlling the programmed cell death in our system.
But cells, as they age, develop problems with their mitochondria. Tell us about the kind of problems that they deal with, and therefore why you need to do some repairs.
Yes. The mechanisms that convert the food that we consume to energy also generate byproducts. These byproducts, in turn, result in impacting the mitochondrial DNA that's in these organelles. So one thing to point out here is that mitochondria are the only organelles that come with their own DNA, in addition to the nuclear DNA that we have, and the replication machinery for these mitochondrial DNA is somewhat primitive, and in addition, the repair mechanisms that they employ is also not as optimal. So as a cascade of events here, the food that you consume and the energy that's produced and the byproducts of that result in accumulating mutations in these mitochondrial DNA. And as a contradictory measure, the organelle doesn't have very good repair mechanisms to fix these.
Okay, that is a problem.
Yes.
Now,
yes.
How might we fix it? You actually have 3 different strategies we're using for that.
Yes.
So let's start with the one you've been working on the longest. We call that allotopic expression.
Yes. Because the repair mechanisms are not as optimal in the organelle itself, we think the best way to approach this problem is by translocating. There are just 13 genes that are vital, that are encoded in the mitochondrial DNA, and they all assemble into the OXPHOS complexes.
So this is the machinery inside the mitochondria that actually does the work of converting energy
yes.
from food into energy to our cells.
Yes, absolutely.
So these are like the components in an assembly line yes. where you need all the parts and to assemble together yes. so that they can actually do the work that the factory is supposed to do.
Yes.
And the genes that are in the mitochondria yes. are responsible for making yes. some of those parts of the assembly line.
Yes. The mitochondrial organelle itself is composed of 1,400 proteins, which deep in the evolution was actually made by the organelle itself. And over time, it transferred all these genes to the nucleus, retaining only these 13 genes that are all parts of the electron transport chain and the oxidative phosphorylation machinery. So our idea is that if we are able to transfer these 13 genes to the nuclear genome and adapt them to the nuclear cytosolic machinery, then we will be able to overcome this cascade of mitochondrial mutation generation and mitochondrial dysfunction as a result of it.
So just spell that out. What's the advantage to having those genes in the nucleus instead of having them in the mitochondria?
Yes. Number one, the mutation rate for the mitochondrial DNA is 10 times more than the nuclear DNA over the lifespan of an individual. This has been shown by several researchers now. The other thing is that the mitochondrial repair machinery is much more rudimentary than the nuclear machinery. The nuclear, so therefore the DNA synthesis and repair mechanisms are much more advanced in the nucleus, and we want to take advantage of this. So once we transfer these genes to the nuclear DNA and adapt them to the transcription and translation there, they can be maintained for a long time and produce these proteins continuously for incorporation into the mitochondria. So there are two other strategies that we have initiated recently. One is called the Gene Drive project, where we are trying to modulate the mtDNA composition of this mitochondria. And the other one is mitophagy. You might have heard about mitophagy recently everywhere in the research field, but what we are trying to do is that we are trying to specifically address this mitophagy from an energy sensing point of view, like the ATP sensing point of view.
So just to spell that out, so there's already this process in our cells called mitophagy that a lot of our listeners will have heard about.
Yeah.
And it is a process that turns over mitochondria successfully, replaces dysfunctional mitochondria with functional mitochondria.
Yeah.
But there is a peculiarity about mitochondria that bear these large deletions that makes them not susceptible to regular mitophagy.
Right. So the influence of these large mutations, in fact, is refractory to this mitophagy process in the sense that they mask these mitochondria from being deleted or turned over in the cells. So as a result, over time, especially in cells that are reliant on mitochondrial function over a length of a time, like the skeletal muscle cells and the neurons, they are differentiated completely, and they don't have the capability of cell division to dilute these mitochondria out. So but they are reliant on mitochondrial function for much of their survival. So you have a lot of dysfunctional mitochondria sitting within these cells. This has been shown for several age-related diseases such as Alzheimer's, Parkinson's, and sarcopenia, which is losing muscle strength grip strength over a period of time. What we think is that if we can address the ATP content, or if we can take advantage of the energy sensing part of the mitochondria and promote mitophagy in just those dysfunctional mitochondria, we may be able to rejuvenate these post-mitotic cells, so-called post-mitotic cells, to bring back energy into them.
Right. So normally there is this masking process, as you said, where the normal destruction and turnover of old mitochondria and replacement with new mitochondria unfortunately misses the most important mitochondria to get rid of.
Right.
Whereas you are working with a different system that senses the energy status of the mitochondria and therefore can selectively turn over the mitochondria that we actually want to destroy.
Yes.
So that's a clever approach.
Yeah.
And then you mentioned briefly the gene drive process, but let's talk about that just a little bit more.
Yeah.
How is that different from the other approaches that we have taken?
So recently there is a lot of research around mitochondria replacement therapy. Many researchers have shown that you can transfer mitochondria isolated from one source into another, but nobody really knows what is the long-term effects of such transplanted mitochondria. But what we are trying to do here is that we are trying to engineer the mitochondrial DNA to reflect certain properties. I cannot tell you all the details because it's a very new project here. But what we are trying to do is we are trying to engineer the mitochondrial DNA in such a way that we can distinguish from the pre-existing DNA, the empty DNA that's supposedly damaged. And when we bring back mitochondria with these engineered DNA, we are developing methods how we can eliminate the
dysfunctional mitochondria.
Yes. That's the approach we are taking. Yeah.
Right. So, how are things going?
Things are going great. As you know, from the Autotopic project point of view, early on we showed that for using a concept called codon optimization. Codon optimization has been used in the industry before, but nobody employed it in a systematic way for mitochondrial DNA. What we showed is that codon optimizing these 13 genes, we were able to show transient expression for almost all 13 genes, and they all associated with mitochondria.
So what we mean there is that we can temporarily cause these proteins to be produced in the main body of the cell, and they get transferred over to the mitochondria.
Yes.
But
not all 13 genes regain function to the level that we want. Some of them did, and for one of the genes, for ATP8, what we showed is that we were able to prove robust expression, import. It goes and assembles into the ATP synthase complex, and it brought back several functions that went missing or absent in a patient cell line. This patient could not make the ATP8 protein at all, so it's what we call a null mutant. And when we replaced a nuclear ATP8 in this patient cell lines, it was able to bring back several functions.
So you got this success with the cell model, and then you've taken that a big step further. You have actually now used the allotopically expressed ATP8, and you have shown that you can actually deliver that into the cells of a mouse. Tell us about that.
Yes. So what we did is we achieved this in a mouse model called the empty FEB mouse. The empty FEB mouse has a point mutation in the ATP8 gene. This is a milder mutation. It's not a null mutation as in the patient cell lines. But what we could show is that this allotopic ATP8 gene, which is now placed in the nucleus in the mouse cells, is robustly expressed, and it assembles very well into the ATP synthase complex.
And it's doing this all across the entire mouse.
Yes.
Right? So this is not just like you've got a few cells here and there. You've shown this in the brain, in the liver, in the kidney.
Yes, all the tissues, it was expressed in all the tissues. And what we have shown that this gene is carried over for at least 4 generations. So, with the progeny, it is carried over. So it's in the nuclear genome, but it is carried over for 4 generations. Right. So we followed these mice for over a year now, and the protein is expressed very, very well, even after 1 year.
And it shows some initial sign that it is actually improving the function of the cells, which is really important because in the real world, people are going to have different kinds of mutations in the cells. And you want to be able to actually, because they're going to be potentially competing with each other. And you want the healthy one to at least do as well as the mutant one.
So that's one very important hypothesis and confirmation that we wanted to establish, that if our allotopic proteins can compete with pre-existing proteins within the mitochondria. And this is going to be a challenge always. So the age-related accumulation of mutations throughout the mitochondrial DNA is going to be heterogeneous. It can vary from person to person, but they are going to produce those mutant proteins as well. Not all of them are going to be null. Often the null mutations are often there is a way to get rid of the null mutations if it is within the cell's repair machinery. But these milder point mutations that influence, such as sarcopenia or a reduction in muscle strength, they are always going to have to compete with mutant proteins. And this is one thing that we show here, is that yes, this allotopic ATP8 can robustly compete with the mitochondrial protein.
And the basis of the whole project was a separate project that we had worked on independently years before, exactly to enable us to carry out a study like this now.
Yes.
This is the so-called Mighty Mouse.
Yes.
Tell us about that.
Yes. This Mighty Mouse is actually was actually developed by Applied Stem Cells. SENS actually funded this project several years ago, and we took advantage of the system in that we can place a single copy of the gene at a very, very defined place within the nucleus, which has been engineered for genes to be expressed very well over time, and it also has the capability of accepting large payloads. So
these are two classic big problems
Yes.
with gene therapy, right? Where on the one hand, you can have a gene that goes in a place that is disruptive to the
Yes.
rest of the genome and causes mutations.
Right.
You don't have that problem.
Yeah.
It can get integrated temporarily, but then the transgene can be
Yes.
lost over time, so
Yes.
you don't have the expression anymore.
Right.
You deal with that problem.
Yeah.
And typically, the delivery systems you use for these
Yes.
genes can only handle very, very small genes,
Yes.
which is not going to do the job for all the proteins we need.
For all the proteins.
Whereas here, this can take both one really large gene, and you can actually take a complex that has multiple genes all together and deliver
Absolutely.
them all together into the cell.
Yes. And from you can now control the copy number.
Mm-hmm.
You can control the sequence of the genes that you are going to express. Yes, this is a very nice model for us, and we took advantage of that. So we use the this is called the safe harbor system, and we implemented the safe harbor system in the empty FVB mice now. Yeah.
So this is how our research is built on itself. So
Yes.
we're already building the model so to be ready for the day when your allotopic expression was ready, and then we were able to bring the two together and actually
Right.
start to rescue function in the mice.
Yes.
So that is incredibly exciting. Now we just have to do that for the other 13.
Yes. We are working on that.
Tell us about some of the other genes, how they're coming along, and which ones are so far working pretty well, and what do you think are the ways you're going to solve the problems with the ones that are not working so well?
Some of the other genes that are working well in our hands is the ND4 gene, which has been implicated in lawn disease, which usually strikes men over the age of 40. The other genes that is working in our hands is the ND4 gene. We are also working on COX2, how we can, so as I said, some genes are expressed very well, and they are also targeted to the right complex in our hands, but some others are not. And we are working on improving the engineering aspects, how we can get these other genes to express very well and target to the right complex. And one of the genes from that point of view we are working now is COX2. We observed that even though COX2 protein is expressed very well, we did not observe a functional recovery for this protein. So we are implementing several other engineering strategies from the protein composition point of view and its targeting elements, how we can overcome that.
One challenge that you have faced is that in order to develop cell models, you need to have cells that have relevant mutations that you can target, because it's no good just putting an allotopically expressed gene into a perfectly healthy cell because it's already perfectly healthy and you can't rescue it. So what kind of tools are available otherwise that you might take advantage of that you can't take advantage of?
So this is a challenge in the field. Primarily, we get our model lines from patients, actually. So
patients with inborn mutations into these
yes,
different genes.
Yes, inborn mutations in the mitochondrial DNA, and for laboratory purposes, we create something called hybrid cells. And we have acquired some of these single gene mutations, let's say, but we don't have for all 13 genes.
And you would normally be able to take advantage of the technology to make mutant cell lines, but not in this case?
Unlike the nuclear DNA editing technologies that has, grown tremendously over the past few years using CRISPR-Cas9 technologies, the mitochondrial DNA is not very amenable to these technologies. In fact, the organelle is double membraned, as you all know, and the inner membrane of the mitochondria is very selective to the molecules it will take in. So with this limitation, this is why we cannot edit the mitochondrial DNA to our will as we want so far. There are some technologies happening, but it's not quite there yet. Yeah.
So you have a dual problem, right? Yes. So you can't just go in directly and fix the problem inside the mitochondria, and if you want to develop a mutant line to test it in, you also can't just easily go in and develop a mutant line as well.
Absolutely. For knockout the number of genes that we want, maybe complex dependent, because there are, let's say, 2 genes in the ATP synthase complex that comes from the mitochondrial DNA. So we can try to knock out ATP8 and ATP6 and now try to express this allotopically and see how it recovers function.
Because as we were saying earlier, you don't just get one mutation, no. You can get 2 or 3 mutations, and sometimes you get these large deletions that knock out all of them at the same time.
Yes.
So you would like to be able to say, can I rescue ATP8 and COX-2 at the same time? Can I rescue ATP8 and ATP6 at the same time? And so forth and so on.
Yes.
Okay, so very good progress, but still more to go.
Yes, lots of work to do. Yes.
Good luck and Godspeed.
Yeah, thank you.